Hello everybody, my name is Taija Mäkinen, I'm from Uppsala, and I would first like to thank the organizers for giving me the opportunity to present our work in this symposium. My lab is interested in understanding the mechanisms that regulate lymphatic vascular development and lymphatic diseases, and in this short presentation I will show you work that we have done on a mouse model of Pik3ca-driven lymphatic pathologies. So here I showed schematically different genetic lymphatic diseases discussed earlier during this meeting, which include both hereditary and somatic diseases characterized by defects in lymphatic vessel formation or their function. And I will focus on diseases that are caused by somatic mutations in Pik3ca gene, and these include cystic lymphatic malformations and GLA. As you know, lymphatic malformations can be further subdivided into macrocystic and microcystic lymphatic malformations. So these macrocystic lymphatic malformations are characterized by large cysts mainly localized into the neck region, while the microcystic lymphatic malformations are more infiltrative and thus more difficult to treat. And GLA is characterized by similar lesions but often involve multiple different tissues. So Pik3ca encodes the catalytic B110 alpha subunit of PI3 kinase that is activated downstream of growth factor receptors and leads to activation of AKT and downstream signaling pathway including mTOR, a regulation of multiple different cellular processes. And mutations in these genes are also commonly found in cancer and several overgrowth syndromes called PROS. And common to these mutations found in these diseases is that they cause constitutive activation of the B110 alpha and therefore chronic or oncogenic activation of the PI3 kinase pathway. And of course, you know about rapamycin that has been used in the clinic now for treatment of these patients due to its ability to inhibit mTOR. And although rapamycin has shown promising results in the clinic, it rarely induces regression of the lesions, and therefore we still need better treatment for these patients. To investigate the mechanisms of Pik3ca-driven lymphatic malformations, we generated a mouse model that allows lymphatic endothelial-specific activation of the common causative mutation Pik3CH1047R. So we use this allele, which can be activated when crossed together with the tamoxifen-inducible CRE line, in this case VEGF receptor 3 driven CRE, and then upon tamoxifen induction, this stop cassette is removed, which allows then expression of the mutant Pik3ca in the cell type of interest, in our case lymphatic endothelial cells. So since these malformations are usually congenital, we first administered tamoxifen to activate expression of this mutant Pik3ca in embryonic vasculature by administering at embryonic day 11. And when we analyzed the embryonic skin at embryonic day 17, we found these large cysts that were mainly localized in the cervical region of the skin, reminiscent of human macrocystic lymphatic malformations. However, when we administered 4-hydroxytamoxifen either late during embryonic development or early postnatal development, as shown here, we observed completely different type of lesions shown here in the postnatal mouse ear skin that were characterized by this hyperbranched vascular network. And histological examination of these two types of lesions confirmed their distinct morphological characteristics. So here you can see just control skin on top, and here in the embryonic skin we can see these large cysts with large lumens, while in this postnatal skin we can see these multiple little lumens, really reminiscent of human macrocystic or microcystic lymphatic malformations that we also characterized together with or in collaboration with Pik3ca. So these results suggest that the developmental timing of P110 alpha PI3 kinase activation determines the subtype of lymphatic malformations. To have a model for testing of different therapeutic approaches, we further developed a highly reproducible model of progressive microcystic lymphatic malformations. And in this model, we induce the expression of the mutant Pik3ca by administering tamoxifen topically into the mouse ear skin at 3 weeks of age. And then we can follow here in the ear skin this progressive overgrowth of lymphatic vasculature. Based on a number of observations from cultured cells and from this mouse model, characterization of this mouse model that I don't have time to show you, we hypothesize that during this progressive growth, signaling via the major lymphangiogenic growth factor receptor 3 may be critical. And so to test this, we inhibited VEGF-C signaling using adeno-associated viral vectors encoding VEGF-C trap. So in this molecule, there is an extracellular ligand binding domain of the VEGF receptor 3 fused to IgG-Fc domain, which binds the available VEGF-C to prevent it from signaling through cell surface receptors on lymphatic endothelial cells. And to our surprise, treatment with this VEGF-C trap led to a very efficient inhibition of the growth of the lymphatic malformations. And why this was indeed surprising is that despite lymphatic endothelial autonomous oncogenic PI3 kinase signaling, the growth of these lesions still depend on the upstream growth factor signaling. So we wanted to then further test the therapeutic potential of this finding. So we compared the effect of this VEGF-C trap to treatment with mTOR inhibitor rapamycin. And for this purpose, we also wanted to treat more advanced lesions to have a more clinically relevant model. So in this case, we again induced the overgrowth at 3 weeks of age and then started the treatment at 5 weeks of age. As you can see here at this stage, the overgrowth is already rather prominent. And so we first tested the effect of rapamycin. For a 1.5 week treatment, we found that we can efficiently inhibit lymphatic endothelial proliferation, which you can see here compared to vehicle-treated mice. And we could reduce the growth of these lesions when comparing here to the endpoint analysis of a vehicle-treated mouse, but we could not see any regression of the vasculature compared to the starting point. When we administered the soluble VEGF-C trap, we saw more efficient inhibition of lymphatic vascular growth, and importantly, the combined treatment with the VEGF-C and VEGF-C trap and rapamycin led to a very efficient inhibition and even regression of the vascular network from the starting point. So here I show you just the quantification of these results, so we can, with the combination treatment, further reduce lymphatic endothelial proliferation and lymphatic vessel branching, even to lower level compared to the starting point. And interestingly, the remaining areas of the hyperbranched lymphatic vasculature in mice treated with this combination treatment show this blunted morphology of vessel sprouts compared to the spiky appearance of actively sprouting lymphatic vessels. So together this suggests that the progressive growth of these microcystic lymphatic malformations is dependent on VEGF-C induced sprouting, which provides a target for treatment. So just to summarize, I showed you that this common causative Pik3ca mutation underlies both microcystic and macrocystic lymphatic malformations, with the developmental timing of PI3 kinase activation determining the subtype. Also showed you that these Pik3ca-driven lymphatic malformations grow in a VEGF-C dependent manner, and therefore the best therapeutic outcome can be achieved by co-inhibition of the upstream VEGF-C and downstream mTOR pathways. So the concept that I would like to bring up is that apart from targeting the lymphatic endothelial cell autonomous PI3 kinase signaling with the rapamycin or PI3 kinase inhibitors, we also need to think about the parenchyme communication of the mutant cells with their normal neighbors, as well as stromal cells, such as macrophages that are important source of VEGF-C that we have shown here can be targeted and provide inhibition of this pathological lymphatic vessel growth. So finally, I would like to thank the people who have done the work. So what I presented has been done mainly by Ines Martinez-Corral, Yen Sang with the help of Milena, and also all our collaborators and funding, and of course you for attention.